Okay. I will probably start with a story. 10 years ago, when I had an entrepreneurial break in my career, we were talking about that before with Daniel. Um, I had to hire a new collaborator. So it was a whole hiring process with candidates. And I had one candidate I interviewed, and she was a fantastic statistician. And I exposed what our mission was in the company. And our mission was to improve reproducibility and help researchers with that transparency, with the replicability of their research. And she was flabbergasted. And she said, why on earth would anybody try to reproduce somebody else's paper? That says a lot. That says a lot. She spent 10 years before in preclinical science, in labs, in biology labs. So that's the kind of spirit we are exposed to in preclinical sciences. So I will try not to depict that pessimistic scenario and an ecosystem now, but we will have to keep in mind that that's pretty much the spirit of many of our colleagues. So I will just have a three steps demonstration and talking first about very quickly the nature of the problem, and secondly I will step into two kinds of solutions, bottom up and top down and bottom up solutions to try to improve the situation, to remedy the situation. I will egocentrically talk about my small contribution, keeping in mind that many people do the same thing, maybe better than me. But I will illustrate all these things by showing what I have been doing over the past 10 years in Lausanne. So that's the kind of statistical paragraph we can find in publications. I got this week in a very recent paper in oncology that used rats and mice. So you can see that it gives an apparent, for a lay audience, an apparent demonstration of transparency here. I bet you can't reproduce this protocol. There are many information that are missing, many information. And if you browse the paper, it's the same, and Valentin will like that. There are no exact p-values, only stars and asterisks everywhere. Many informations are missing here. So my mistake, they disclose the software, at least. But all the rest is just missing. So exact sample size, you don't know. Randomization, you don't know. Blinding, you don't know. All these things, you have no idea if they did, and if they did, how they did it. So that's the nature of the problem, and that's everywhere in preclinical sciences. Because researchers, they are just not aware of the importance of this information. Why is that a problem? Well, we are facing various issues here. The first issue is ethical. The use of animals is not just using free resources like consumables. They are animals, and the right we have to use them, the right we decide we have to use them, is intricated with our duty to replace, reduce, and refine their use. That's the 3R that we are compelled to comply with in animal sciences. Of course, we waste other resources, money, etc., etc. But there are also more profound issues here, which are more epistemological. It jeopardizes the value of our science, of our research endeavor to increase science, our scientific knowledge. And it jeopardizes the credibility we have in the global community beyond the scientific community. So it's more important than just having one paper reproduced. It's more widespread. It's a more widespread philosophical problem we are exposed to. So there are three major aspects, three major remedies we can try to implement to improve the situation. I expose them like this: guidelines, requirements, and education. So guidelines are more recommendations. We may be free or not to adhere to them, but there are recommendations. Requirements are enforced recommendations, so there are rules. Researchers usually don't really like rules. They are kind of free thinkers, but they don't like rules. Education. So we all took statistical lectures when we were undergrad students. Then we forgot everything, and suddenly we start a PhD and we have to do this again, and we forgot everything beforehand. So education is key, and the state, the year, the curriculum you choose to give this lecture is also important. But education in terms of curriculum during your studies is just one part of the study, of the story. We can also educate researchers by education papers and statistical papers in the literature. We are going to talk about that later. Let's start with guidelines. So guidelines, they are not new. They've been there for decades or even more. As we discussed before, medical guidelines in terms of statistics, they've been there for ages. More seriously since the Declaration of Helsinki in 1948. So the medical community is forced to be transparent in their use of research and in how they implement medical research. In the preclinical world, these guidelines, they took ages to take root. They came much later. And for decades, what we had were that sort of guidelines, a lot of text about how to handle, how to anesthetize animals, how we should use the recovery time, etc., etc., etc. Maybe you saw some text that was highlighted in yellow. That's the only part of the guidelines dedicated to the fundamental criterion is to give a description that would allow the study to be replicated by a similarly trained research worker. That's all. So for ages, that's the only thing we had. And in parallel, there were statistical guidelines for clinicians, not restricting their use to clinical studies. You could use them if you were not a clinician, but there were completely separate guidelines. Now, guidelines exist, mostly clinical guidelines. They exist, they are updated and they are gathered here and available in the Equator Network website. And I will, of course, talk about the most important, almost the only guidelines that exist for preclinical sciences, for the use of animals. It's the ARRIVE guidelines. So the ARRIVE guidelines, they were published first in 2010, I think, and they were updated in 2020. They are important guidelines. They bring two novelties. First, they were published, you can see, in PLOS Biology, and they are somewhere between guidelines and requirements, because basically, if you want to publish in PLOS Biology with experimental animals, you are forced to follow the guidelines. So there are guidelines, but there are also requirements. Now you are free to choose another paper, another journal that does not adhere to the ARRIVE guidelines, if you wish, like the one that I presented in the very first slide of my talk. The second thing is these guidelines, they dedicate a large part of their content to statistics, a large part. Design, analysis, presentation, the test you use, what sort of design you had, the cross design, factorial design, what tests you used, what exact sample size you had, the blinding, randomization, etc., etc. All these things must be disclosed somewhere, somehow, which is pretty new. It's pretty unique still in the world of animal sciences. Now the ARRIVE guidelines are quite complex in terms of their contents. You have a lot of items you should comply with. And they are also associated with some problems. Guidelines are only guidelines. And it's been shown for other types of guidelines that if you don't really enforce something, you always have a way to skip the guideline. And Douglas Curran Everett, which is a prominent statistician, let's say advocate for correct education, published in 2004 guidelines for reporting statistics in journals published by the American Physiological Society. Interestingly, they also published a sequel, so the follow-up analysis of the impact of their guidelines 3 years later. So what they found basically is that their guidelines had absolutely no effect. So that's concerning in two ways. First, minor concern, the authors, they don't care. So we expected that. We expected that researchers might not read the guidelines. But what's more concerning is that the editors of these journals, which all published these guidelines were published in every single journal of the American Physiological Society. The editors, they don't care either, because basically they just let papers go without enforcing the guidelines. So you see the issue with guidelines, they are just mere recommendations. So I started to get interested into that issue a couple of years ago, and I used a scoping review approach, just to have a clear picture of what's the use of statistics in the preclinical world. What exactly is are the current customs in terms of statistics in the preclinical world, in people working with animals, biochemistry, molecular biology, you name it, molecular physiology, cell physiology, all these fields that appear upstream to clinical science. What's the use of statistics? So in this scoping review here, I analyzed a lot of papers, just looking at how transparent they were in terms of statistics, their design, but also the reporting of their results. I said I tried to not to be too pessimistic, but sometimes the results, they speak for themselves. What you see here is that the existence of flawed presentation or incomplete presentation, bad presentation, or the absence of presentation of statistics, statistical methods, or statistical results is quite prevalent. On the left-hand side here, you have 20 journals. So each dot is a journal, and you have the percentage of papers from this journal that show the flow that you see underneath. Some journals, apparently, they think okay for their papers, for 80% of their papers, not to disclose the test, the statistical test they use. How on earth would you like, could you expect to replicate that results if you don't even know what kind of analysis they did? Same for the sample size, you have no idea. Undisclosed software. The software is not trivial. Sometimes maybe you use R, maybe you use GraphPad Prism, SPSS, etc. All this software, they don't compute the p-values the same way. If you do a simple t-test, R will use a Welsh correction automatically by default, which is not the case for other software. So the result, the output you will have will differ. This is not a trivial information. So you can see, and importantly, because sometimes I get this comment from researchers, from students, it's pretty much independent from the impact factor. So it's everywhere. Okay. This is concerning all the more because guidelines are not new. The concern about statistical reporting is not new. Valentin, you mentioned history before. You mentioned some kind of a timeline. Fisher himself, so the father of modern statistics, he warned us 1 century ago. He said that biologists, they don't know how to handle statistics and we should teach them So it's not new. And about the clinical, or about the statistical significance, he also said that he personally, and he used the word personally, prefers to set the significance threshold at 2 standard deviation, but this should be changed study by study, by the way. So this is something that everybody forgets. History repeats. So I kept doing that kind of studies in specific, very specific fields. In this case, I just illustrate some specific studies I did in the world of immunoblotting. So Western blotting, for those of you who are not aware, it's the use of antibodies to detect and quantify proteins in samples, biochemistry, pure biochemistry, very used in, for example, in oncology. You have Western blots everywhere. And I found pretty much the same thing wherever I looked at. Unclear tests, unclear or undisclosed sample size, etc., etc. So I started to think, if guidelines are not enough, why not try to enforce new rules? Why not? Why not, after all? And I started to write IDs. And by writing IDs, I ended up with manuscript, which I sent to BioSS. And I managed to get it published after discussing with the editor-in-chief. It's just a bunch of ideas I had about what could we do in the future? What could we try to do to change the awareness, to change the way preclinical scientists consider and report statistics. My idea was that by changing the way we enforce manuscript writing, we might change the mindset upstream. So why not standardize the contents of the statistical paragraph with mandatory items? Why not make the statistical paragraph the first paragraph of the method section? After all, it makes sense. You all start by designing a research, which is part of the statistical process. Why not insist on the existence of a paragraph that lists and comments and discuss the limitations of your study, not the biological or conceptual limitations, but the statistical limitation. You use the parametric test, why? What are the limitations? Your sample size is quite small or too large. When you have a very large sample, you may be confronted with the Linless paradox that makes your frequentist results in contradiction with your Bayesian conclusions. So all these things might be discussed in a dedicated paragraph that should be mandatory. And also journals should allocate resources, space to studies that work on reproducibility itself or publish null results, etc., etc. All these things could be enforced in the scientific community. Maybe it was just a stone thrown out of the sea, but anyway, I tried. Education, probably the most exciting thing I try to do, and we probably all try to do, teaching statistics. I am also a biologist. I am not a statistician. We have statisticians here. I am a biologist. I'm a self-taught statistician. So I do teach statistics. I teach statistics to PhD students, and I've been doing that for more than 10 years now. But I do teach statistics without mathematics. That's the trick, which requires me to know the mathematics. Usually I do lecture series of several days. I focus on very introductory and applied approaches. I try to tackle misconceptions about p-values, about testing, about sample size, about misuse of all these things. It's probably more important for biologists to know how not to use statistics rather than how to use them, considering the extent of the problem. Now, teaching statistics is probably not enough. This is clearly a bottom-up approach. I've been called a couple of times by students to attend their lab meetings, because whatever I can teach them, whatever the solution I can give them in the lecture hall, they're all confronted to supervisors who say, okay, that's a nice story for your studies, but now I want my p-value significance now. Okay, you want to use a very nice statistical test? I want the t-test to be done. What can they do? Except calling me to attend the lab meeting and say, no, not really. That's how it should be done. So there was always this problem with education, with bottom-up education. So I said, oh, maybe we could try to also to educate the researchers themselves. So forget the seminars and the education, the formal education that doesn't work with researchers. There is a glass ceiling that you can break after the PhD. So I had this idea last year to just to increase the statistical education in the community of animal researchers or researchers on animals. And we have this idea. I take the, well, I benefit here from the fact that I am a section editor in statistics from the laboratory animal journal. So it was easy for me to come up with this idea of having a special issue with a collection of articles specifically dedicated to statistics for applied researchers in animal science. We had quite a success, and it was a kind of overwhelming for us. I don't know if some of you are editors, or if you have editorial activities. You know how painful it is to find a reviewer for one paper. Imagine for 20 or 25 papers. It was quite painful, but it's rewarding at the end, because it's going to be a good success. I think we will have around 20 articles. Each of these articles will cover a very small article, very small article, will cover a specific statistical issue dedicated to animal science. So we expect the publication of the issue pretty soon, in summer, or maybe late summer. But I think it's going to be a very nice resource to communicate to animal researchers. So I hope I was not too pessimistic. I am realistic and fatalistic as well. There is a problem of awareness in the field of preclinical science. The researchers, they are not really aware of the importance of a right transparency, the good communication. They think that the only important thing is just the results they present without everything upstream. That's a real issue, and we really try to change things. Hopefully it will change, but who knows? Who knows? I'm still an optimistic guy. So I would like to thank you for your attention, and for the question that will ensue. Thank you very much.
Thanks a lot. Do we have questions? Yep.
So you said you are trying to solve the problem by teaching stats to non-stats folks. And you teach a lot them how not to do statistics, but they still need to do statistics. How do you suggest that they actually go about that when they need to, because they do?
Yeah, the contents of the lectures quite they are complex in terms of how they hinge around the concept of statistics. Mm-hmm. I always introduce the statistical concepts, and then usually quite directly I switch to the misuse and misconception. There are a lot of practical activities as well. Quiz is very easy. very easy quiz. How to not badly interpret the error bars, for example. And I found that this approach is very educational, and they understand quite well the theory by starting with the misconception. So then after, I communicate some materials, PDFs, etc., to dig a little bit more into the fundamental knowledge if they wish. That's pretty much the approach. But also that's an introductory lecture that also aimed to prepare them for more advanced lecture they would undergo later on for multivariate analysis or software exploration. It's really an introduction to the future lecture they might take.
Do you know whether they do or whether they do stats themselves? Do you know whether they do take more advanced courses, whether they do statistics
Yes.
themselves or they reach out to someone?
Yeah. I would say that half of them do. Those who need the lectures, for example, those who work with the omics data or complicated psychology, behavioral, neurobehavioral, or neuroimaging, they do take courses, more advanced courses. But the course I give here did not exist before, so there was a gap between their absence of knowledge and the advanced lectures they had to take. This introductory gap, I tried to fill it, and I found it quite useful, and they found it quite useful as well. So because now they can start with the very basic theory of probabilities, p-values, testing, just understanding all these things before going to do some PCA or whatever. That's an important step that we managed to do.
Absolutely. I'm just from personal experience working among non-stats, not mathematical people who do research. I've seen cases like, oh, I didn't use that method because it used something called linear mixed model. That's so complex. I just used a t-test instead. And you're like, that's not appropriate. Oh, it's too complex. I won't use it. Sorry. And they managed to publish nevertheless. And that's kind of, you cannot do it because also reviewers would accept it. So that's.
No, but you get easily lost in translation.
Yes.
Even for concepts they think, or we all think we master because we hear about it all the time. My little baby those days is the normality assumption. We all know, we all think we know, we all think we know how to test it. Actually, we don't. The exact nature of this assumption, the exact testing of this assumption, the exact nitty-gritty of how it's somewhere in statistics is quite obscure. It's a black box for everybody in biology. So it's Claude Bernard, the physiologist, who said that it's what you already know that prevents you from learning. That's exactly that. Everybody thinks they know something, and that prevents them from going further into the knowledge. Being lost in translation is something that I try to break with these lectures. After the lectures, these biological students, they are not lost in translation anymore when they talk to statisticians, at least.
Nice. Thanks for great work.
Okay, super. I think we go on with the next presentation. So thanks a lot, Romain-Daniel. It was great. Yes. Thanks a lot. So we quickly make a small here break again. So we stopped.